I now introduce the next speaker of the session, and this is Brian Taubenheim. Brian holds a bachelor degree in chemistry and a master's degree in business administration. And he started his professional career in the CDMO industry, then transitioned into pharmaceuticals, holding roles in CMC development, operations management, business development, and therapeutic program lead. He is, since 2022, the Chief Operations Officer of Bellhaven Biopharma. And he'll talk about a life-saving epinephrine product and the impact of particle size and patient factors on nasal deposition. So over to you.
Okay. Thank you. Thank you for the introduction. I'd like to thank the organizers and DDL for having us. I'll get a few themes out of the way. We also use the UDSP device, so Aptar is getting good bang for their buck on their sponsoring today in these first three sessions. So that's a carryover.
Could you step a little closer to the microphone?
Oh, can you not? Here, let's do this. Is that better? Yeah? Okay. So lawyers like to have us start with a lot of words. In short, we're going to do our best interpretation of the data that we've collected and tell you about our research today. So as we see in these presentations and in the poster sessions, the nasal administration is well established and growing. We can see that by following the dollars, and the cash really tells the story. So over the next 6 years, you see the compound annual growth rate in the intranasal space is set to grow at 6.6%. And interestingly, the dry powder delivery device systems are slightly larger projected growth at 7.5%. The drivers are really the technology. So again, as you saw at this conference, there's ever increasing technology on how we deliver, how we produce, how we analyze, and how we produce successful products. Patient preference is driven by self-administration. So people don't want to do needles. That's obvious. There's a large growing population of elderly, and the outpatient treatment of those elderly patients is also contributing to the interest in intranasal delivery. And then there's the expanding the access, so the ease of delivery and also the ease of distribution through the normal pharmacy channels to get drugs out into the population. Important in the regulatory timeline, especially to Nasdepi, was the first approval of a dry powder nasal, that being Baqsimi, in 2019. So that was developed to treat the diabetic population for hypoglycemia. A few months ago, ARS Pharma produced Neffy, a liquid nasal epinephrine for anaphylaxis. And those really serve as our analogs and help guide us on our regulatory path for Nasdepi. So before we spend money and do cool science, we like to ask ourselves, are we doing something that's going to ultimately provide patient benefit? So we use the abilities as our guide. So first, is it going to be effective? In the case of Nasdepi, we're looking at bioavailability and comparing that to the already approved therapies on the market. Is it going to be usable? So as we've heard, the UDSP device is very usable. It's already been proven in the market, and there's easy dose administration. Reproducibility, are we going to be able to scale this up, make it reproducible, and we want to design a big design space if possible so that we can go into manufacturing and have devices come out, work at the same time every batch as long as the product's on the market. So we need to be able to store it. Yesterday, if you happen to walk around the poster session, we talked about our stability. No surprise from the speakers you heard earlier, dry powder nasal formulations are quite stable. So it's effective, it's reliable. People carry these things around in their pockets. They throw them in their glove boxes. They take them to the beach. They put them in their jackets when they go to the mountains skiing. And all those scenarios, you want to be able to pull that device out when you need it and have it work. And then distributability. So are we able to get this to the patients? Minimal controls are needed for the shipping and moving of the product around the world, as well as, again, a compact deliverable device that can get to the patient. And ultimately, it needs to be approvable. So using the precedent analogs I just mentioned, as well as having a close collaboration with FDA, we summarize all these abilities into a regulatory acceptable package that, again, ultimately provides patient benefit. So in these two studies, we examined the impact of the spray-dried drug and particle size. So the objective was to demonstrate the formulation deposits primarily in the nasal cavity with minimal lung availability. We used two powders in this, SDD1, which had a smaller overall distribution, and then SDD2, which was about 3 times as large. In study A, we assessed the impact of the device filling method. So we looked at a Quantos filler, we looked at a Harrow tabletop drum filler, and on the Harrow, we looked at applying the vacuum at a low setting of 200 millibars and high setting of 600 millibars and see if we get any compaction. And ultimately, again, how does that impact what comes out and what gets administered to the patient? In study B, we looked at usability and bioavailability. So we used a model and we looked at different orientations, horizontal and vertical, airflow, no airflow. We looked at different coatings with 12 CP and 80 CP, again, to assess as we develop our product, we use it with this particular device. Ultimately, how does it serve the patient? So we used a common method throughout the two studies to measure the emitted particle size distribution using a spray tech. Study A had actuation with the Proveris into the model, and we used the Alberta idealized nasal inlet model from Copley. And in study B, we had a manual actuation, and we also measured the aerodynamic particle size, and we used the Aeronose model developed by Aptar. So in study A, our hypothesis was that the PSD and filling process, both the equipment and the vacuum, should impact the emitted particle size distribution. We did indeed see a difference in the equipment filling, where we used the smaller particle size with the Quantos and the larger particle size powder in the Harrow tabletop drum. And as you can see here, the blue line, I think there's a pointer. The blue line is the difference with the smaller particle size. With the two pressures, we used the same powder, the SDD2, with a larger particle size. And as you can see, there was no difference between a low vacuum and a high vacuum setting. We suspect that the smaller particle size had increased cohesive forces, specifically van der Waals and capillary forces, and that contributed to the larger EPSD we saw. And also, we saw that the compaction that we suspected from the vacuum setting really had no difference when it came to the EPSD measurement. We took those filled units and went to the ANI model, and here we hypothesized that the smaller EPSD and larger fine particulate fractions should increase the lung exposure, so make it past the model towards the filter. Powders showed that there were no significant differences between the preparation method and EPSD that reported results up to 17% fine particulate fraction in the model translated to really less than 5% of the powder when passed and was available to the lungs. So in conclusion, PSD and EPSD are really not good predictors for the possible lung deposition. And what we do, taking that back to our development, is we spent a lot of time trying to optimize and get to the sweet spot, or what we thought was the sweet spot of the bulk particle size, when really you should be focused on how does the powder perform in the device early on versus trying to engineer your powder before you get to the device. In study B, we looked at the impact of the position of the model, nasal coating, airflow, all those on deposition and in combination. We thought, hypothesis coming in again, something would be different when you start changing all these variables. We were wrong. So all the results, you saw 74 to 84% of the total drug emitted out of the device went into the nasal cavity, into the tubernates, which was our targeted area. Airflow did have an impact, significant impact, though not a impact that's going to be significant to a patient, where we saw the less than 1% jump to about 5% as far as lung availability, potentially. But positioning, horizontal, vertical, different coatings within the nasal mucus or nasal mucosa, all resulted in very similar results with less than 1% being available to the lung. So in conclusion, the variables studied really did not have a meaningful impact of the powder for the projected absorption. So in summary, excuse me, SDD with a DV50 of less than 5 microns may have an impact on EPSD, again, through those cohesive forces. However, the bulk particle size distribution and the translated post-device emitted EPSD are not accurate for predicting potential lung exposure when compared to the anatomical model. We did introduce the APSD. It was closer to the anatomical model, but it may indicate that it actually underestimates the potential for lung exposure. The studies ended up showing us that we had a wide range of filling operation parameters that were feasible without impacting deposition, and also good for the patients. It seems from the models that patient positioning, whether you have a runny nose or you don't have a runny nose, whether you're inhaling, you're conscious or unconscious, you should be able, through the device and the powder, get a consistent administration of the drug that's bioavailable. All these studies added to the support of the program with formulation design in conjunction with the UDSP device will reliably deliver the drug to the patient when they need it to the target areas of the nasal cavity. I'd like to thank my 2 lead collaborators, Gonzalo Ferreras from Aptar and Matt Owen from Catalent. I believe they're in the audience, and we are happy to answer any questions.
Thank you very much for this interesting talk. While we are waiting, there is already a question over there. Go ahead.
Brian, really great talk. Philip Keel from Loveless Biomedical.
Hi.
Hey, I wanted to ask real quick, what do you think your package insert is going to be relative to airflow? And how do you factor that into what you do for your, and I get it, it's in vitro testing. It's not meant to be truly predictive. But do you think there's a correlation there? And do you think you're going to do it with a breath hold, or do you think you envision doing with it a little bit of an inhalation?
We're hoping there's no requirement. So what we're shooting for is, again, as easy as it can be for the patient. So even though 15 liters, you saw normal respiration airflow, you still saw a good distribution of the powder within the targeted areas. And when you didn't have any airflow, so an unconscious patient, which for anaphylaxis is possible, you still got a good distribution. So we'll test both those, but ultimately we're hoping there's nothing in the label about what type of respirations you have to have going on.
Thank you.
Mm-hmm.
As it is an emergency medication, I guess a quick onset of action is wanted. Do you have any idea how fast your powder will dissolve? So what is the time to peak max?
So we presented our first clinical study data a few months ago, and we saw it very comparable or faster to the current treatments with the EpiPen, either the auto-injector as well as the manual injection. So the dissolution's quick and the systemic bioavailability is faster or equivalent to. One interesting point, we did see prolonged drug exposures, so accumulation stayed, and we had a longer half-life than the intramuscular injection, which could be interesting when you start talking about breakthrough reactions. Mm-hmm.
So I don't see any other immediate. Oh, there is one. Sorry.
Everybody wants refreshment break.
A question, it's out of curiosity. I guess to some extent it doesn't matter if it gets into the lung, given that one of the manifestations of anaphylaxis is bronchospasm. and adrenaline counteracts bronchospasm as well as the other systemic effects it has.
Yeah, I think you're correct. It's really not a safety issue. It's more of a consistency in the PK exposure. So getting it in the same place all the time versus you get different PK absorptions if you're getting into the lung.
Just I have a question. Generally, the nasal cast which we are having in the market, they are generally prepared by using any of the plastics or some other models. But in the realistic scenario, we are failing, means there is no mucus coating done to the nasal cast. So do you seriously see any impact on the data which you have projected?
So if I understand the question was the impact of the nasal coating?
The materials used for the nasal cast, they are generally plastic or any other materials. But for the current scenario, in the realistic scenario, what do you say? There is some mucus layer and adhesive layer. They are generally observed in the nasals. So there is quite difference in the realistic as well as the in vitro tools which are available. So do you see any of the difference in the deposition patterns or any impact?
Yeah, so that'd be a great question for Copley. We didn't design the model, we used the model as they prescribed. I did see a poster actually right next to ours yesterday that talks about how the different coatings, you can get various, variability in your outputs. But we didn't study that specifically, so we used it as prescribed.
Thank you very much for your talk. It was great information. I was just curious for understanding, like you mentioned that nasal XX provaris, so I was just trying to understand how are the behavior in a spray pattern or plume geometry.
So I had a hard time hearing the question. Plume geometry?
Could you
Yeah,
repeat?
spray pattern and the plume geometry.
We're looking at plume geometry. Again, we're sort of in new grounds. We're being forced down the regulatory path of there's a lot of liquids out there, and plume geometry is sort of a standard when you start talking about the liquids. When you start talking about powders, especially delivered through the UDSP, it's really a bolus of powder. And it hits the nasal mucosa and it sticks, and that's where it is. So we'll do these plume geometry studies. We're getting consistent results, but ultimately we think it has little impact on the efficacy of that we're going to see of the device and the powder combination.
Okay, so let's thank Brian again.